talk about organs of the immune system. I talked about different types of microorganisms that immune system is going to deal with them. So we have different types of immune cells in order to be able to induce different immune response against different types of microorganisms. And you saw this image in the last session that we have different types of cells. Before talking about the cells of the immune system and organs of the immune system, it is important to know that we do not have any immune response that is induced only by one cell. We do not have any immune response that immune response is only is being done or is induced by only one cell. We always have a network of cells that simultaneously they cooperate with each other in order to induce a proper immune response. So always we have a network of cells against, for example, viruses, fungi, bacteria, and other microorganisms. It's very important. And I'll talk about immunologic networks in the, I think tomorrow in the cytokine lecture again. Let's skip these slides. We do not have enough time. We have different cells, you know all of them. I'm going to talk about them one by one. I talked about overview of the immune system now. So, what is this image? What is this image? Where is that come from? Where is that image is taken from?
Slide for blood. Slide for blood and
Yes, it's a blood Espirande slide, which is stained with hematoxylin. And you saw different, so they are blood cells. Is that correct? They are blood cells. They are indeed cells of the blood. We have erythrocytes and platelets, eosinophils, monocytes, neutrophils, lymphocytes, basophils, and erythrocytes or RBC. Do RBCs have any role in immune system? Do RBCs have any role in immune system or not? I'll talk about it. One important role of, yes, they have. They of course have a role. Yes, erythrocytes or RBCs have one important role in immune system. I'll talk about it in complement section tomorrow. So, yes, clearance of immune complexes. They take the immune complexes with themselves to the spleen, and inside the spleens, the immune complexes are or beaten by macrophages. So, let's go back to the lecture. Cells of the immune system. We have different, so all immune cells, except we can say most of them, are indeed the cells of the blood. The cells that they circulate in the blood circulation. They migrate from different parts of the body through blood circulation. And you know that monocytes, after going to the tissues, differentiate to macrophage. You know this, that monocyte is indeed ancestor of macrophage. And macrophage, we do not have macrophage in the blood. In the blood we have just monocytes. But when monocytes migrate to the tissue, inside the tissue, they differentiate to macrophages. So, the blood cells, we have three main types of blood cells: RBCs, or red blood or erythrocytes, you can see here; white blood cells, or leukocytes, or immune cells. White blood cells, another name for white blood cells is leukocytes. You know that red blood cells have hemoglobin, and hemoglobin is red, and in hematoxin they are reddish. So red blood cells, the color comes from that, but white blood cells do not have hemoglobin, so they are not red. And and platelets. Let me skip. It's important, you are not going to memorize the content of this table, but it is important to know that we do not have equal numbers of all cells inside in the blood circulation. You know this. That, for example, we have 5 million in average RBCs in a cubic millimeter, but for the rest of the cells, for some of them we have thousands of them, for some of them we have few of them. For example, basophil, we have only 1% of the leukocytes in the blood circulation is the basophil. But the most frequent cells of the white disease or leukocytes in the blood circulation is for neutrophil. 50 to 70% of the white disease. These percents are the percent of each cell from the white disease, not all cells. I mean, they are not included for red blood cells and platelets. So, 50 to 70% of white disease of the blood circulation is neutrophil. Then we have 20 to 40 percent for lymphocytes, and then monocytes 2 to 12%, and so on. So some of them are very rare. For example, basophils. Mast cells are usually a tissue resident cell, and the most abundant is for neutrophil. And the lifespan is again different. For example, for neutrophil, the lifespan of a neutrophil is 6 hours to 2 days at the maximum. But for example, for macrophage or monocyte, we have days to months. And if they differentiate monocytes, if differentiate to macrophages, they can live for years, sometimes, and sometimes they can live for decades in the tissues. And memory cells, lymphocytes. It's important to know that both B lymphocytes and T lymphocytes, as well as NK cells, are counted here. This percent or this number include T lymphocytes, B lymphocytes, and NK cells. So, from the previous image, this percentage comes from the morphology of the phenotype of the cells. But you know, you are not going to memorize them, but they have the appearance is different totally. For example, monocytes that do not have granule, but eosinophil, basophil, and neutrophil have some granules, or lymphocytes are small cells. Both T cells, B cells, and NK cells are same in the appearance, not the function. I'm talking about the appearance, morphology, or phenotype of the cells. So we have different number of each cells inside in the blood circulation, and their lifespan is different as well. But you are not, and this is called. There is a test in the laboratory which is called CBC, for complete blood count. In complete blood count, we have the exact number of red blood cells, platelets, and leukocytes, and their percentage. And any changes from this normal range, those ranges are called normal ranges for each one. Any changes from those normal ranges could be a sign of a disease. For example, when we have a higher number of lymphocytes, maybe it's, of course, it should be interpreted beside the other laboratory test. But the higher number of lymphocytes shows a viral infection, or the higher number of neutrophils show a bacterial infection. It is important to keep in your mind, and you know, I think, most of them. What is hematopoiesis? No. We have hematopoietic stem cell, that's right. What does production and development of blood cells, including RBCs, platelets, and leukocytes, or immune cells, or white blood cells. You know that all of the blood cells comes from one stem cell, which is called hematopoietic stem cell. These stem cells can produce all cells of the blood, including white disease, platelets, and RBCs. And you know that that before birth, the production of blood begins in the yolk sac and then in the liver and spleen before birth. And in the birth, the bone marrow is the main site of blood production. So before birth, the main site for blood production is liver, spleen, and yolk sac. But after birth, the different bone marrows. And in a young person, mainly tibial, and long bones are the main site of blood production. But in an adult, the blood production is mainly done in the, for example, ribs and other parts of the body. I think you know most of them. Am I right? You know those
Yes.
things.
Yes.
That before birth in the yolk sac, liver, and then bone marrow, and different bone marrows from childhood to adulthood. And this is a hematopoietic stem cell that can produce all the cells. You can see here that we have two branches of the cells. One branch is this one, the other branch is this one. The upper branch, which is called myeloid lineage cells, all of these cells are myeloid cells. Indeed, they are, all of them, they are developed from a progenitor, myeloid progenitor. That's why, for example, you know some, you know this cancer, CML. Or ALL. They are types of blood cancer. This is a lymphocytic cancer. This is a myelocytic cancer. Chronic myelocytic lymphoma or acute lymphocytic lymphoma. So I wanted to say to you that we have two branches. The lower branch is indeed lymphoid progenitors. That this lymphoid progenitor can produce T cells as a lymphocyte, B cells as a lymphocyte, besides NK cells and ILCs. ILCs are a specific type of innate immune cells indeed, but they have some properties of innate immune cells and some properties of lymphoid adaptive immune cells. So the lower branch are lymphoid progenitors or lymphoid cells. Lymphoid cells are mainly T cells, B cells, and NK cells. The upper branch are myeloid cells that all of them develop from myeloid progenitor that include erythrocytes, platelet, and rest of the leukocytes, including mast cell, basophil, eosinophil, dendritic cell, monocyte, and neutrophil. So these kind of leukocytes are called myeloid cells. Myeloid cells. So when we say myeloid cells of the blood, we mean those cells. Is that clear? Because their progenitor is a myeloid progenitor. But erythrocyte and megakaryocyte comes from myeloid cells as well. Is that clear? I'm trying to summarize as much as possible. So myeloid branch, including erythrocyte, platelet, and non-lymphoid cells. Granulocytes, monocyte, dendritic cells, and all of them are indeed they are descended from a common myeloid precursor or progenitor. And lymphoid progenitor and lymphoid cells, I talked about them, and they are called lymphoid lineage as well. So now I'm going to talk about different cells of the immune system in summary. I'll try to talk about them very, very, very shortly as much as possible. So you know that we have many types of cells with different morphology and different appearance and different functions as well. And we have different categories between them. For example, we have neutrophil, eosinophil, and basophil. All of them are called neutrophil, eosinophil and basophil. Those three cells are called granulocytes because they have granule all of them inside them. Granulocytes. And again, macrophage, macrophage, neutrophil, and dendritic cell. They are called macrophages, neutrophils, and dendritic cells. What they are called together?
Phagocytosis.
Phagocytic cells. They do phagocytosis mainly. No, monocyte cannot do. Monocyte is a precursor of macrophages. Monocytes do not phagocyte anything. Monocytes convert to macrophage and then they can phagocyte extracellular antigens. And we have a group of cells which they are called APCs, antigen presenting cells. What type of cells they are? They present antigens to T cells. Which type of those cells are APC? Dendritic cell and B cell. Let me find. A small lymphocyte, for example, B cell and macrophages. Yes, they are the main APCs of the body. So all of them are in this image. APCs, granulocytes, and phagocytic cells. No question? No question? So, first of all, I'll talk about neutrophil. What's the main function of neutrophil? It is the most frequent cell of the blood. They are produced in bone marrow. We have a very good pool of neutrophil in the bone marrow. Whenever the body needs more neutrophil, they can release from bone marrow. So, what are the main functions of neutrophil? Phagocytosis, yes. Phagocytosis and degranulation. Inside the neutrophils, there are some granules. Let me back to the previous slide. You can see that we have some granules inside the neutrophils, eosinophils, and basophils. Why they are called neutrophil, basophil, and eosinophil? Because the content of the granules is different. They have granules, but the content of their granules different from each other. And in the staining, eosinophils mainly is stained with acidic or eosine stain, but the basophils are stained with basic stains. And neutrophil is the granule of neutrophil is stained with neutral stains. Is that clear? So the content of the granules is different, so they absorb different kind of stains. Neutrophils absorb neutral stains, eosinophil absorb eosine as an acidic stain, and basophil absorb basic stain. So the names come from the content and the staining of the granules. And inside the granules, they have some content that those contents are harmful for their for example, eosinophil has some granules that the content of granules is harmful or toxic for worms. And inside the neutrophils, there are some enzymes and some proteins that they are toxic to, for example, extracellular bacteria or fungi. And inside the basophil, there are some other toxic materials that they are toxic to worms, for example. So they are like bullets of the cells. Whenever they need the bullet, they can release the content of bullet to the target cells, for example, worms, bacteria, or fungi. So indeed, they are the tools for them to use them against the target. So, neutrophil. One function of neutrophil was phagocytosis. Another function of neutrophil is degranulation. It means whenever neutrophil cannot phagocyte its target, it releases the content of the granule on the target cell. If the target is small, for example, a bacteria or a fungi, a bacterium or fungi, neutrophil can phagocytose them. But if the target cell is a big cell and if neutrophil cannot phagocyte that cell, it will release the content of its granule on the target cell. And I told you the content of these granules is toxic to the target cell. For example, they have some enzymes, including lysosome and some peptides, which they are called antimicrobial peptides, and some enzymes, and some other products that they are toxic to the target cell. There are another function for neutrophil which is called netosis. In the netosis, we have a programmed this of the neutrophil. Neutrophil dies in this function and release the content of its all the contents of its granules besides nucleic acids. You know that we have nucleic acid inside the nucleus of the neutrophil. Neutrophil will release its nucleic acids alongside with the content of granules and make a trap in it. A trap which is composed of nucleic acid and toxic materials that by this way, this trap can trap, for example, a bacteria or a bunch of bacteria or a bunch of fungi. Is that clear? It is called netosis. You might have heard about apoptosis. Yes. Another program said this. This one causes comes from apoptosis. But we have neutrophil extracellular trap. This net is indeed neutrophil extracellular trap. Neutrophil makes a trap having so many toxic agents against bacteria and fungi. No question? So neutrophil has mainly three functions: phagocytosis, degranulation, and netosis. If we have whenever we have a chronic inflammation, you know that neutrophil is one of the main cells of inflammation. Okay? Neutrophil is one important cell of the inflammation. Whenever we have inflammation, inside the inflammation site we have many numbers of neutrophils. But whenever we have chronic inflammation, you know that chronic inflammation is harmful for our cells. Why chronic inflammation is harmful for our cells? By knowing these functions of neutrophil. When we have chronic inflammation, indeed we have chronic activation of neutrophil, chronic degranulation, and chronic netosis, and these granules and these traps can harm normal cells as well. When we have acute inflammation, they fight with microbes, and some normal cells may die, it doesn't matter. But when we have chronic inflammation, they can kill so many normal cells. And our tissues will be damaged by the neutrophils and of course other cells. So chronic inflammation is harmful because a neutrophil can harm the cell tissues, normal cells, normal tissues. No question?
Neutrophil and chronic inflammation will decrease the number of neutrophil and chronic inflammation.
Your question again?
Chronic inflammation.
Okay.
The number of neutrophil will decrease, sorry.
Inside the blood or site of the inflammation?
Blood.
No. We have a pool of neutrophil inside the bone marrow that when we have a chronic inflammation, there are some cytokines produced here, and the cytokines go to the bone marrow, and indeed those cytokines say to the bone marrow that we need more neutrophils. And neutrophils are released from the bone marrow and they move toward the chronic inflammation site. So we have more numbers of neutrophils in the blood circulation, not the lower number. Another question? No? Nothing? So another cell is eosinophil. It's an anti-parasitic cell. You know, it's important against parasites or worms. And the content of its granules is harmful to the parasites. Another cell, basophil and mast cells. They are similar cells, but basophil is mainly found in the circulation, in the normal situation, but mast cell is mainly in the connective tissue. We have some different numbers and different types of even mast cells in the connective tissue. Both of them are involved in the initiation of inflammation. In the triggering of inflammation, they are important.
Because it's releases histamine.
Histamine is, yes, one of its content is histamine. I'll talk about the content of granular mast cell and basophil in hypersensitivity or allergy lecture. And they are important in, they are involved in allergic reaction. Besides eosinophils. Eosinophil, basophil, the number of them will increase in the allergic reaction in the blood circulation because they are involved in allergic reactions. You know this. No question? And you know that in allergy, we have one antibody which is called Ig. Which type of Ig immunoglobulins is important in allergy? IgE. And IgE can trigger the function of basophil, eosinophil, and eosinophil. Basophil, mast cell, and eosinophils. So all of them are involved in allergic reaction and anti-parasitic reaction or immune response. Another important cell is very important is monocytes and macrophages that I told you. Monocyte is a blood cell. We do not have monocyte inside the tissues. When monocytes go to the tissue, inside the tissue they differentiate to macrophage. And indeed the monocyte is ancestor of macrophage. And we have two main types of macrophages that I'll tell you in the next lecture. But the main function of macrophage, tell me, what are the functions of macrophage? Phagocytosis, yes. What else? Antigen presenting cells, yes.
Cytokine.
Production of cytokine and initiation of inflammation, you mean? Yes, initiation of inflammatory response. Yes, thank you. So, macrophage is important, a phagocytic cell, and besides, it's important in tissue repair. I'll tell you about it, because macrophage is very important in tissue repair, because they can produce some growth factor and angiogenic factors that they are involved in the remodeling of the tissue after tissue damage. You know that one important function of innate immunity is tissue remodeling or tissue repair. And tissue repair, macrophage is one important function of macrophage. And they are important in inflammation. They are by production of cytokines, they induce inflammation or inflammatory response. And they are an APC cell or antigen presenting cell. Question, comment? We have different types of macrophage in different tissues. I think you know about some of them from your histology course, I think. For example, in brain, we have they are called tissue-specific macrophages. It means that they are specific to some specific tissues. For example, microglia are indeed a macrophage. That this macrophage is a tissue-resident macrophage of brain. They are only in the brain, which is called microglia. In the lung, we have alveolar macrophage. In the liver, we have Kupffer cell. Kupffer cell is another macrophage. Kidney-resident macrophage. We have Kupffer cell only in the kidney, not other tissues. Or in the kidney, we have kidney macrophages. We have Langerhans cells in the skin, another tissue-resident macrophages, and other. So it is important to keep in your mind that monocytes are in the blood and they migrate to the tissue and they stay in the tissue. They do not go back to the blood. And they are called tissue resident macrophages. The most important of them are microglia, alveolar cells, Kupffer, and Langerhans cells. They are indeed macrophages or tissue-specific macrophages. Their function is same. All of them initiate inflammation. All of them can do APC. All of them do phagocytosis. Another important cell of innate immunity. All of the cells until now I've told you were the cells of innate immunity. Granulocytes, monocyte macrophage, and now dendritic cell. What is the or are the functions of dendritic cell? Antigen presentation, yes. Phagocytosis and production of cytokine to induce inflammation. It's similar to the macrophage, but dendritic cell is much stronger APC compared to macrophage. But macrophage is much stronger phagocytic cells compared to the dendritic cell. Both of them have the same functions, but the APC function of dendritic cell is much stronger and phagocytic function of macrophage is much stronger compared to each other. It is important to know. So phagocytosis and induction of inflammation, and it is said that dendritic cell is the main bridge between innate and adaptive immune system. I told you that innate immune system or innate immune cells active or activate the adaptive immunity. And one important cell of the innate immunity that activates adaptive immunity is dendritic cell. It is the main bridge, one of the main bridges between innate immunity and adaptive immunity. So those cells are called professional phagocytes, neutrophil, dendritic cell, and macrophages. But you know that B cells can do phagocytosis because they are an important APC, and eosinophil can do phagocytosis. That's why we say they are called professional phagocytes. So the professional phagocytes are those three cells, but we have other phagocytic cells as well, including eosinophil and B cells. I talked about granulocytes. Another important cell is natural killer cells, NK cells. What are the functions of NK cells? NK cell? Yes, they can induce apoptosis. When they induce apoptosis? Yes, they are important in viral infection and in tumor immune response. They are very important cells of innate immunity and very potent cell of innate immunity against viral infections and tumor cells, because indeed they are against mainly intracellular antigens. They induce apoptosis in the viral infected cells. They induce apoptosis in the tumor or cancerous cells. So they are important in transmembrane. Yes. The content of NK cells, there are two main types of proteins in the granules of the NK cells. NK cells have granule inside them, and inside the granule there are two proteins, including perforin and granzyme. When they release the content of the granule on the target cell, you can see them here. When they release the content of the granule in the target cell, on the membrane of target cell, perforin will make a pore or hole, and granzyme is an enzyme that goes inside the cell, and inside the cell it induces apoptosis. Okay? NK cells can become activated with different mechanisms, including, so again, The main function of the NK cells was induction of immune response against intracellular antigen, mainly viral antigens and tumors. And now I'm going to talk about that how an NK cell become activated. How can we activate NK cell? NK cell become activated with different mechanisms, including ADCC. I'm not going to talk about it now. I'll talk about it in antibody and human immune response and in other lectures. I'll talk about this. This is activation of NK cell by antibody. You might remember some thing about it. Another way by which an NK cell can become activated is this, that on the membrane of NK cells there are some receptors, that those receptors can recognize some normal and abnormal self-antigens. For example, in cancer we have two types of antigens: normal antigens, cancerous and abnormal antigens, cancerous antigens indeed, and normal antigens. NK cell have a receptor for both of them. If on the membrane of a cell there are abnormal antigens, which sometimes they are called stress proteins, any cells that undergo to a stress condition, for example, any stress, or infectious stress or tumor stress, any kind of stress, they will express stress proteins. Those stress proteins are not normal. They are not expressed on the normal cells. And NK cell can detect this stress protein. And NK cell will find out that, oh, this cell is abnormal, I have to kill it, by detection of the stress proteins. When a cell is under a stress condition, it should be eliminated, it should be deleted from the normal cells. So NK cell do this by recognition of those stress proteins or those abnormal proteins on the cells. Okay? For example, if we have a cancerous cells, in a tumor cells, we have some antigens that they are not expressed in a normal cells. NK cell can detect these normal cells, abnormal cell, and kill them. But the mechanisms of the killing in all mechanisms, in all ways, is same: releasing of perforin and granzyme and induction of apoptosis.
Doctor, can you repeat that?
The second one, you know, NK cell can discriminate between normal and abnormal cells. Normal and abnormal cell cells. How can discriminate NK cell between normal and abnormal cells? In the outer membrane of an abnormal cell, there are some proteins that they are called stress proteins. They are not expressed on the membrane of normal cells. For example, this one is a normal cell. It has some normal antigens on its surface. And this one is an abnormal cell. It has again some normal antigens and some abnormal antigens. The red ones are abnormal antigens. And NK cell can recognize these abnormal antigens. So NK cell when see these abnormal antigens will understand that this cell is abnormal and then become activated and release the content of its granule on the abnormal cell, but not on the normal cell, because on the surface of normal cell there is no abnormal antigen. Clear?
So it has a receptor for the normal and the abnormal cell.
Both of them, yes.
Yeah, it's activated
When only it recognizes normal antigens, it will do nothing. It says that this cell is normal. But when it recognizes abnormal, besides the normal, it will understand that this cell is abnormal. Oh, I have to kill it. And it will kill it by releasing of the content of its granules. Okay? There is another mechanism by which NK cell become activated, which is called absence of MHC. I'll talk about it in the next lectures. There are three different mechanisms by which NK cell can become activated. Absence of MHC, ADCC, and recognition of abnormal or stress proteins on the abnormal cells. Self cells, I'm talking about self cells. Do you know anything about absence of MHC? What is MHC? Major histocompatibility complex. What's the function of MHC? Present the antigen to T cells. Thank you. So what is the absence of MHC? Prevent the expression of MHC. Some viruses and tumor cells try to prevent the expression of MHC in order to evade from immune system, in order to become recognized by T cells. So tumor cells and some viral infected cells try to prevent the expression of MHC in order to escape from T cell response, to escape from cytotoxic T cell response. But immune system have NK cells for those cells. If you, for example, immune system says to the tumor and viral infected cells, okay, if you do not express MHC, I will consider you as an abnormal cell, and I will kill you. NK cell says to the tumor cells or viral infected cell. Is that clear? So we have indeed a competition between tumor and immune cell, for example. Tumor tries to suppress the expression of MHC in order to escape from T cell response. On the other hand, immune system will arise another cell, NK cell. That NK cell can kill any cells that do not have MHC. You know that MHC, all nucleated cells have MHC on their surface. So whenever there is no MHC, it means again abnormality. In normal condition, we have MHC on the surface of all nucleated cells, all of them. When there is no MHC, it's again a sign of abnormality. And NK cell is sensitive for abnormality again. And we recognize that cells and again we kill the target cell by perforin and bronzine. One question? So NK cell is very important, very potent cell in immune response against viral infection and tumors. And lymphocytes as a cells of adaptive immunity. I talked to you that we have different type of lymphocytes. I'll talk about them in the adaptive immunity. So, those type of cells in different stages of the cells in the lifespan, from naive cells, activated, effector, and memory. You should know all of them. When we have no, when a lymphocyte hasn't recognized its specific antigen, it is called naive cells. I told you. But after recognition of antigen, it is called activated. This activated lymphocyte will differentiate and proliferate to an effector cells. As the name shows, and a naive cells is a naive cells. It cannot do anything against, it cannot fight with the target cell. But an effector cell, for example, plasma cell, will produce antibody. That antibody will fight with the target antigen. Or T cells, for example, cytotoxic T cells, before, when they are naive T cells, they cannot kill viral infected cells. But after they differentiate to effector cells, they can kill the viral infected cells. I'm talking about CTLs. So effector cells are the cells that have capability to fight with the target cell or fight with the microorganism. And some of the naive cells, after recognition of antigen, will differentiate, or some of those activated cells will differentiate to memory cells. Memory cells, you know that they can memorize the experience with the antigen, and in the next exposure, they will induce secondary immune response, a stronger and faster immune response. And memory cells, after bonding with antigen, again, they will differentiate to effector cells. both of them. So memory cells, after recognition of antigen, they will differentiate to effector cells again. But there is a difference with these effector cells and these effector cells. What is the difference between those effector cells? Effector cells, the question is this. We have two types of effector cells. Effector cells that come from a naive cell and effector cells that come from a memory cell. What is the difference between them?
That one is more powerful than
More?
stronger. More powerful.
Yes, the number is, they can induce a stronger immune response because we have a higher number of effector cells here. We have a huge proliferation of memory cells here. And they can induce immune response faster than previous one. Okay, question? And in the lifetime of a person, you can see here that over time, with the age, the number of naive T cells are decreasing, and expectedly, the number of memory T cells are increasing. What does that mean? It means that over time, a person is experiencing antigens, the more number of antigens. Okay? For example, when a 10 years old child, for example, it has the naive cells are more than the memory cells because it hasn't exposed to many antigens. But over time, for example, in a 30 years old man or woman, the number of naive T cells and memory cells is equal because they have exposed to more antigens. And over time, here, for example, a 70 years old, the number of naives are less than the number of memory cells because they have experienced more antigens. Any exposure to any antigen will produce new memory cells. I'm talking about different antigens. So expectedly, a person with higher age has exposed to more antigens, more different antigens. When it is exposed to more antigens, more different antigens, so it has more memory cells. Is that clear? Over time, during the lifetime of a person, the memory cells are increasing and the number of T cells decreases over time. Is that clear? So what do you know about cluster of differentiation? CD markers. They are called CD markers. CD markers. On the cell membrane, surfaces of the cells, cell membranes, you mean? Yes? What do you know about CD markers? You've heard about them. Can you tell me what number of CD numbers have you heard until now?
4,
CD4, CD8, CD3,
45,
19,
45,
CD45,
56,
and so on. So what they are? They are in these CD markers or cluster of differentiation is a nomenclature or naming system used to name different proteins or different macromolecules, not only proteins, any macro, glycoproteins, carbohydrates, that they are expressed on the cells. There is a naming system for that. And we use this naming system to differentiate between cells. For example, we know that any cells that have CD3, it's indeed a lymphocyte. And any cells that have CD3 and CD8 is a cytotoxic T lymphocyte. Any cells that have CD3 and CD4, it is indeed a T helper cells. And CD45 is a CD marker for all leukocytes. You know this. I'm telling you that we have a naming system for different types of macromolecules with different functions. We have a naming system to use them to discriminate different cells from each other. For example, all granulocyte has CD45, and monocytes has CD45 and CD14. T lymphocytes CD45 and CD3. But T helper cells, CD45, a marker of leukocytic cells, and CD3, a marker of lymphocytes, and CD4, a marker of T helper cells. It's a numbering, it's a naming system. And cytotoxic T lymphocyte, CD45, a marker of leukocytic cells, and CD3, a marker of lymphocytic cells, and CD8, a marker of cytotoxic T cells. And B lymphocytes, CD45, a marker of leukocytic cells, and CD19, a marker of B cells, and so on. You are not going to memorize all of them. Whenever you need to talk to memorize any CD numbers, I'll tell you. I'm telling you here that we have CD markers as a naming system for different proteins that they are expressed on the different cells. So we use these CD markers to discriminate or differentiate the different cells from each other in the research or in the clinical laboratory. And they are used widely in both research system and in clinical laboratory to distinguish different cells from each other. Question, comment? So, have a break, 10 minutes break, and then I'll resume lymphoid tissues.
الاسم اللي راح اصيحه معناته بسجلته غياب تمام؟
Bone marrow and thymus. Yes, bone marrow is the site for indeed the central lymphoid tissues are the tissues that immune cells are produced or developed there. All leukocytes and blood cells are produced bone marrow except for T cells that they are produced in the thymus. Even precursors precursor of T cells developed from hematopoietic stem cells of the bone marrow. So all leukocytes of the blood cells are arise from hematopoietic stem cells in the bone marrow. All of the cells develop and mature inside the bone marrow, but the precursor of thymus leave the bone marrow and goes to the thymus, and inside the thymus we have the development and production of the T cells. So we do not have foreign antigen exposure in the central lymphoid organs. We do not have any foreign antigen exposure in the primary lymphoid organs. But in the secondary lymphoid organs, that we have different types of secondary lymphoid organs, including adenoids, tonsils, lymph nodes, spleen, and so on. We have hundreds of lymph nodes around the body, and all of them are called secondary lymphoid organs. The main function of secondary lymphoid organ is the function of secondary lymphoid organ. No. The maturation is for the primary lymphoid organs. Initiation of immune response. Beginning of immune response. Adaptive immune response. So the development of cells occurs in the central lymphoid tissue, but the initiation of immune response occurs in the secondary lymphoid tissue. It is. So, we do not have any exposure to foreign antigens here inside the thymus and bone marrow because the cells are not mature. We have immature cells that after maturation, the cells are developing inside the thymus and bone marrow. After the maturation, they leave the thymus, T cells, and other leukocytes in the bone marrow. They leave the bone marrow and circulate throughout the body and goes inside the secondary lymphoid organs, and inside the secondary lymphoid organs and throughout the body usually they circulate waiting for antigen. Or it's better to say they always are circulating throughout the body to search for the antigen. They are trying to find the antigen. And the immune response is initiated in the peripheral lymphoid tissue. And we have exposure to foreign antigens in the peripheral lymphoid tissues, not primary lymphoid tissues. You can see here that we have, for example, common lymphoid progenitor. Here is bone marrow. that a lymphoid progenitor, the precursor goes to the thymus for production of T cell and bone marrow for the production of B cells. Not only B cells, the rest of the leukocytes except for T cells, all of them are produced or developed in the bone marrow. And after maturation inside the thymus and bone marrow, they leave the primary lymphoid organs or generative lymphoid organs. They are generation sites for the leukocytes. And they leave the primary lymphoid organs and goes through the circulation, goes to the secondary lymphoid organs. And those organs are the organs that we have initiation of immune response. I talked about hematopoiesis, BSC. You can see here a real smear of the bone marrow on the slide. That they are fat cells and different type of cells that's being produced in the bone marrow. It's a real image. For example, you can see here neutrophils and another neutrophil-like cells, lymphocyte-like cells, and rest of the cells are immature. So inside the bone marrow, when we have a bone marrow, we see a bone marrow smear under the microscope, we will see a heterogeneous population of the cells, including all leukocytes except for T cells and the immature form of them. From, for example, an hematopoietic stem cells to the, for example, mature B cells, T cells, red blood cells, platelets, and rest of the cells. Okay. So, thymus, you know that is in the upper chest behind the sternum, and it has a capsule and a cortex and a medulla. Cortex is the site that is under the capsule, and medulla is the central part of the thymus. And I'll talk about this again in the sorry, in tolerance lecture. This is the part of thymus. This is the capsule of thymus. This part is the subcapsular region or the cortex of the thymus, and here is the medulla. And T cells from the cortex toward the medulla, when we go from the cortex towards the medulla, the cells are more mature. Indeed, and immature cells from here migrate from cortex toward medulla, and step by step it becomes a mature cell. Okay? We have, and it is said that around 95% of immature T cells in the thymus they are eliminated by apoptosis. Only 5% of T cells can leave the thymus. Okay, please, again, only 5% of immature T cells can leave the thymus. It means that only 5% of these cells are normal cells, and 95% of them are eliminated by apoptosis. Why is that? What's the reason behind it?
Because we have a process.
What process? I'll talk about it. We have process of tolerance induction, and we have a random production of TCRs on the T cells that I'll talk about them, that some of them are autoreactive cells, self-reactive cells, and those autoreactive, self-reactive T cells are eliminated by apoptosis, and only 5% of them are not autoreactive or self-reactive T cells, and they can leave the thymus. I'll talk about it in the tolerance lecture. And it is important to know that inside the thymus, those cells are lymphocytes, developing lymphocytes, immature, and for example, this one is immature that can leave the thymus, for example. But we have dendritic cells as well, macrophages. Whenever we have apoptosis, we need macrophage to phagocytose the apoptotic bodies or the cells, the remnants of the dead cells. So we have macrophages in order to apoptose the dead cells, the dead T cells. We have some kind of dendritic cells as an APC, and some specific type of epithelial cells that they are APC here as well. We have two type of APC here, dendritic cell and in some specific type of epithelial cells. Inside the thymus, those APCs are presenting antigen to the developing cells. What does that mean? We have the thymus is full of developing cells that they are proliferating, and again among them we have antigen presenting cells that they are presenting antigens to the developing cells. Why we do need for the antigen presenting cells inside the thymus? What antigen they are presenting? You know antigen presenting cells. They present antigen to T cells. And here they are the T cells. So what kind of antigens are presenting to the T cells inside the thymus? What kind of, yes, the reason is that to prevent autoimmunity. What kind of antigens are presented by them? Pathogen. No. Yes, they have MHC on the self, for example. Self-antigens. They are presenting self-antigens on the MHC to developing T cells. Indeed, they are checking the T cells if they are self-reactive or not. If the T cell is self-reactive, it will be eliminated by apoptosis. And we have some macrophages to phagocytose them. Okay? If when they are exposed to antigens, if the T cells are not self-reactive, they are indeed competent to become mature and leave the thymus. So inside the thymus, we have developing T cells, antigen presenting cells including dendritic cells and epithelial cells, and macrophages in order to phagocytose the dead cells. No question? That's enough for thymus.
Sir, T cells produced in the bone marrow, but measure in the thymus.
What again, please?
T cells produced in the bone marrow.
No, it's precursor, not the T cells. It's progenitor. Indeed, its ancestor comes from bone marrow. For example, a precursor, or for example, this one, for example, is a precursor or progenitor of T cells. But inside the T cells, you have many rounds of proliferation and differentiation towards a mature T cell. Okay? No question, no comment? Both CD4 positive and CD8 positive T cells and regulatory T cells are produced inside the bone marrow, not other cells. Just CD4, CD8, and regulatory T cells. I'll talk about them again. This is a real image. You can see that the dense population of the cells, they are the cortex. Those parts are cortex. Those parts are the medulla. They are the cortex. Here is the medulla. And you can see that in the cortex we have a dense population of the lymphocytes. But in the medulla we do not have a dense population of the lymphocytes, because most of the cells from the cortex that goes towards the medulla, they eliminated by apoptosis. Indeed, most of them were self-reactive T cells and they were eliminated by apoptosis. That I'm gonna talk about them in the tolerance lecture. So it's a real image of the one part of thymus. No question? So lymph node and lymphatic system. What is this?
Capillaries.
Capillaries. In the tissues that we have artery, arteriole, and capillaries. And you know that from the capillaries, plasma exit the blood and go and make the indeed interstitial fluid, and some of the interstitial fluid is absorbed by the venules, and you know that some part of the interstitial fluid is absorbed by the venule, and some other is absorbed by lymphatic vessels. Okay. Lymphatic vessels, the question is this: what is a lymphoid or lymphatic fluid? Lymphatic fluid, can you define it for me? Lymphatic fluid.
Doctor,
How is it made?
it's fluid that's collected with pathogens and
Collected with pathogens? No. When you are not infected with any pathogen, you have lymphoid fluid. It's not related to pathogen. A normal person without any pathogen, without any immune response, has lymphoid fluid. It is collected from interstitial tissue fluid. You know, yes. Here we have an artery that takes the blood to the tissue. Those are the cells of the tissue, and from the capillaries we have the exit of plasma. To the exit of plasma from the capillaries makes the interstitial fluid that bathes the tissue of the cells of the tissue, that feed the tissue of tissue cells. That it brings food, brings oxygen to the cells of the tissue. It's the function of blood, and so from the capillary, plasma goes into the interstitial tissue, feed them and reach oxygen to them, and then some part of this interstitial tissue is taken, is absorbed to those green ones are lymphatic vessels, are absorbed to the lymphatic vessels. So lymphatic fluid is indeed very similar to the plasma or interstitial fluid. Okay? Is that clear or not? You know that when blood comes to the tissues, cells are not allowed to leave the capillaries unless in inflammatory situation. Cells routinely, most of the cells, red blood cells at all are not allowed to leave the capillaries, but plasma and some leukocytes, for example monocytes, can leave the capillaries and go into the tissue. For example, here we have interstitial fluid and some leukocytes. So, after feeding the cells of the tissue and bringing oxygen for them, so that the oxygenated plasma and the plasma that has less amount of food has to be absorbed by the venules and veins. It's clear for you, I think. But some part of interstitial fluid is absorbed by the lymphatic vessels, goes into the lymphatic vessels, and even some leukocytes, not all of them. And those lymphatic vessels take the lymphoid fluid to them by themselves toward the lymph nodes. You can see them here. We have those are lymphatic vessels. Throughout the body, we have lymphatic vessels that they take the fluid from the interstitial fluid. Why does we need to take the interstitial fluid to the lymphatic vessels? Why do we need for this process absorbing of the interstitial fluid by the lymphatic vessels that we call it lymphoid fluid? And why would we do need this whole process?
To identify to the foreign things like microorganisms.
In order to sample, indeed, by absorbing some part of interstitial fluid, indeed we are sampling the tissues if there is any antigen or not. How immune system can check the tissues for the presence of, for example, microorganisms? By absorbing of the interstitial fluid that it might have microorganism or not, antigens of foreign antigens or not, by absorbing them or indeed by sampling them and taking the fluid to the nearby lymph nodes. You can see the lymph nodes here. All of them are lymph nodes. And inside the lymph nodes we have a dense population of immune cells, including T cells, B cells, macrophages, and dendritic cells. So, inside the tissue, in order to immune system be able to sample the content of the tissues to check if there is any foreign antigen or not. Okay, we have lymphoid fluid formation or absorption of some fluid of interstitial fluid inside the blood lymphatic vessels and taking it to the lymph nodes. We have hundreds of lymph nodes along the lymphatic vessels. So we have, similar to our blood vessels, we have lymphatic vessels almost all part of the body. Not all part, not exact all of them, almost all of them. And we have to check this, let me, I think I have another. You can see that those vessels are lymphatic vessels. That they are sampling all parts of the body. Indeed, here we have some lymphatic vessels that take some samples, indeed interstitial fluid by themselves, to the nearby lymph node. If there is antigen inside the body tissue, here inside the lymphoid organs or lymph nodes, we will have initiation of immune response, generation of immune response. Is that clear? So always lymphatic vessels are taking some part of interstitial fluid in order to sample the tissues for the presence of foreign antigens or abnormal antigens and take the samples toward the lymph nodes. And finally, you can see here that we have heart, arteries, veins, and lymphatic vessels. The yellow one is the lymphatic vessels. This one is lymph node and this one is lymph node. So inside in the tissues, we have absorption of the lymphatic fluid to sample the presence of antigen here and take the antigen to the lymph node. If there is foreign antigen, we will initiation of immune response. Without having lymphatic system, immune system cannot check the presence of antigen in the tissues. And after initiation of immune response, we will have eradication. And finally, all the lymphatic fluid will go back to the blood from the, I think, left subclavian and right subclavian. You know this from anatomy. So, there is a closed circulation of the blood and lymphatic system. We have both lymphatic vessels and blood vessels throughout the body. So the component of lymphatic fluid is very similar to plasma that has some leukocyte inside it as well. Clear? No question? You know what is lymphatic fluid or not?
Yes.
You know why we need for lymphatic fluid or not?
Yes.
You know how immune system check the presence of antigen inside the tissue or not? Okay? You know? No question? What is the final destination of lymphatic fluid? The final destination of lymphatic fluid, what is?
Lymph nodes.
After lymph node, we have, for example, we have lymphatic fluid, lymph node, then what?
Then it's the heart.
Again lymph node, then what? Again lymph node, then what?
Blood circulation.
Blood.
Heart.
Circulation. All of them finally ends to the subclavian right and one right subclavian and they drain to the blood finally. So they drain the content of the tissues and finally goes to the blood. So there is a closed circulation of blood and lymphatic system. So inside the lymph node again, I'm not going to the details as much as possible. We have some, you can see here, some lymphatic vessels that they are called afferent lymphatic vessels. They bring they bring the lymphatic fluid from the tissue toward the lymph node. And we have just one efferent lymph node, several afferent lymphatic vessels and one efferent lymphatic vessel. This one, those one, bring the lymphoid fluid toward the lymph node, and this one take the lymphoid fluid from it drains the lymph node and take the lymphoid fluid to the another. And it is important to know that, for example here, this one is afferent lymph node, for example, and this one is efferent. Or it is important to know that this one is an afferent lymphatic vessel for this lymph node, and this one is efferent. The efferent of one lymph node is afferent of another one because they are alongside of the lymph node. For example, here again, we have an afferent of this one, efferent of this, and afferent for another one. Okay, the lymph nodes are indeed connect different lymph nodes together, and indeed one lymphatic fluid pass through several lymph nodes before getting back to the blood. Why does it happen? It happens because it maximizes the recognition of the antigen by the specific cells. Because inside the lymph nodes we have a dense population of the adaptive immune cells, including T cells and B cells. So when for example an antigen is here, goes to the first lymph node, there is no specific T cell for it. The next lymph node will check it. Again, there was no specific T cells. Inside this lymph node, then there might be a specific T cell. Is that clear for you? So when a lymphatic fluid passes through different lymph nodes, okay, 1, 2, 3, 4, and 5, it maximizes the exposure of antigen, the chance of exposure of antigen to its own specific antigen, specific T cells or B cells. Okay, so afferent lymphatic vessels bring the lymphoid to the lymph node, and efferent take the lymphoid from the lymph node, and inside the lymph node again we have a capsule, and beside the capsule we have a subcapsular region that we have follicles inside here. You can see them here. They are follicles. What do you know about follicles? What do you remember of follicles? Follicles are the site of the presence of B cells. Follicles are called B cell zone. So inside the lymph node, it is important to know that T cells and B cells are not they are not spread inside the lymph node whenever they want. We have a specialized part for B cell and some other specialized part for T cell, which is called T cell zone. You can see here. So we have follicles for B cells, and we have T cell zones underneath the B cell zone. That's enough, I think. I'm not going to talk about the details anymore. No question? So, if there is any antigen, we have B cells here and T cells here. Again, we have dendritic cells here, we have macrophages here. Without APC, we won't have any T cell response. So whenever we have T cells, we must have APC besides them. So we have T cells here and dendritic cells beside them, and we have B cells here again. If there is any antigen, they can activate B cells or T cells and dendritic cells, and we have again macrophages and many other. They are the main cells of the lymph nodes. T cells, B cells, dendritic cells, and macrophages. Is that clear or not? This is the dendritic cells that is taking, I told you, inside the lymphoid fluid, lymphatic fluid, okay? Inside the lymphatic fluid, we have some leukocytes, including dendritic cells. That dendritic cells take the antigen by themselves to the nearby lymph node. You can see here the dendritic cell that is taking the antigen, for example, from the tissue toward the lymph node. And inside the lymph node, this will present antigen to the T cells. And it will activate the T cells. And after activation, these T cells will leave the lymph node and goes to the site of infection. Site of infection. Okay? No question? Spleen. Spleen is very similar, is indeed a big lymph node. Again, it's very, but it's the main site for damaged blood cells and blood-borne antigens. Blood-borne antigens. What does that mean? If any antigens directly go to the blood circulation inside the spleen, the antigens will express to the T cells and B cells again. So it's another secondary lymphoid organ. It's it's the site for the initiation of immune response. So immune response against damaged blood cells and blood-borne antigens. Blood-borne antigen means the antigens that go directly to the inside the blood. And again, inside the spleen we have T cell zone and B cell zone. I'm not going to the details. That's enough. Let me skip. And this slide as well. That's enough for lymphoid organs. So, I'll get back to class in 15 minutes for the next lecture.